Julie describes coffee in the manner of peak. What's peak Julie?
We'll get there together.
All right. So come on out.
All right.
And I guess describe coffee in the manner of peak Julie.
All right. I'm trying to I'm trying to figure out what flavor of Julie to go with here, though. That's the thing. I mean, it's going to require me doing some lying, I think, is the problem. Is that okay?
Oh, yeah.
Okay, cool. So there are many phyla of bacteria. A lot of them are distinguished by, I mean, the major means of distinguishing them and categorizing them is by cell membrane composition, whether that contains or does not contain lipopolysaccharides. But another strong aspect of classification is morphology. There are several different kind of general shapes that bacterial cells may take. Some common ones are rods or spheres. There's some spore-forming ones. There's some spirally ones. There's some weird ones too. But yeah, so we're still working on classifying coffee beans themselves. They're kind of an elongated caucus. They tend to swarm, which is why you see so many of them in a bag. You have to harvest them from moist environments. They are facultative anaerobes, similar to a lot of the coliforms or the fecal bacteria found in humans.
Don't look at the brain. Don't stop ruining coffee for me. Oh.
So there's actually, we've got a long tradition. There's many. I mean, we've commercialized the process now, but there are still artisanal. There are still artisanal coffee caucus gatherers present in some of the damper places of the planet, who have really had the chance to hone their art over the past several centuries that we've been drinking coffee now as a species. So the traditional method of preparing coffee is the caucus harvester will generally dive down. Some of these are deep underground and are partially watered. Think like a cenote, but smellier. So what you do is you have to get right down in there. Technically, you want to get kind of as far into the water column as you can, because they are facultative anaerobes, which means they can grow in an oxygenated environment, but they really prefer a lower oxygen concentration in their surroundings. So you want to get kind of, so the way these geological formations work too, right? And I'm not a geologist, so excuse me if I'm not, using the correct terminology here, but you kind of want to go, sorry, I'm about to do I need to use my hands here for a second. Yeah. So here's the typical coffee caucus cenote is kind of, actually a little colon shaped. So, you'll have your entry down here. The water level's usually around here, and so what you want to do is kind of, the diver will go kind of down, and then the peak harvesting points are usually in the rock formations kind of on the underside. So, if you think of like the ilium, you gotta go down into it, and then that way, kind of into it, and then yeah, just under the rock shelf, and then you kind of get up into the slimy bits, and then that's where you'll find clusters of coffee cocci, generally clinging to the walls. They usually form kind of a slime on the substrate. So you have to gently scrape these off, gently. And there's a few different collection mediums you can use. You want to keep that sealed tight. So generally they'll go into a swab, kind of picture a really long Q-tip. It's what we use for sexual test screening swabs as well. It's just a really long Q-tip. So you get in there, you take a good swab. You can get several million colonies on a single swab. So it's a really economic process. So you get your swab, it's up in there. You want to immediately snap and cap that into a preservative media before you can take that back to the brewery. So you get that, you snap it, you cap it, you're good. You want to get back out of that ilium real quick because there's some gases down there you don't want to be exposed to for too long. Once you're up at the surface, generally you want to immediately wrap that tube in usually we go with just tinfoil in the field. It's cheap and it blocks sunlight because you don't want to expose it to the sunlight for too long. It's pretty susceptible to radiation damage.
Is this still coffee?
Yes, I'm getting to that. So you have to take that back to the lab. as Serge will tell you, the fresher the better, right? So you don't want to, ideally you want to get that growing as soon as possible from the field for peak freshness. So you can freeze it, you can store that at about, I think we usually use about you can use a -60 freezer Celsius, of course, and that'll preserve it for a while. And that's fine, but what you really want to do is get that into an incubator right away, so that you're not halting their growing process. Because again, also the freeze thaw cycle can, especially if they're stored incorrectly, you can get micro spike formation that can impact the flavor, the flavor plasmids. So you don't want that. So ideally, you go straight from the field. Within 12 to 24 hours, you get that into the lab. And you immediately want to start incubating that. And I'm trying to remember the temperature. They are mesophiles. I think you generally go with, again, it depends on the strain. I want to say it's between like 27 to 32 degrees Celsius in an incubator. Again, shielded from light. You want to do this in the dark. It's a dark reaction. But they will, and it's a long reaction because they don't... because of their colonial nature and because of the environment, they're usually in a kind of a restricted environment, so they don't normally grow to the size that we see in stores. Until so that's why you have to incubate them, and we usually do like you can do about 24 to 30. Again, this is really strain dependent. Thank you. Yeah, this is actually really similar to what I wear in the lab. So yeah, about it. So if you do like. I'll go with the more common, like the K19 strain. So that's a 24 to 36 hour incubation at about 29 degrees Celsius. There's some wiggle room, but yeah. So you leave that, overnight and a bit, and when you come back to lesson. You go home, you go sleep, you go do other science, and then you come back in the morning, and hopefully, if everything's, sometimes they can be finicky, but hopefully you come back. And they will have approximately, so another thing that's interesting to note, and this is just one side info, but when you think of a coffee bean, that's actually not a singular organism, that's actually a colony. So when you grow these overnight, they start to, you start from a film, right? You start from a thinly streaked film that you can't really see. But through the incubation process, they start to, they really, once they're kind of settled, there's a lag phase, so you don't see growth right away. But then they get attenuated. They start taking up nutrients from the medium. They start reacting to the heat, which is quite a bit warmer than their natural environment. So they really like that, and they'll start forming colonies. And those, so they double. Like other bacteria, they reproduce by binary fission. You can actually see kind of the line of fission. The groove in the bean itself, that's pronounced on the roasting. So yeah, so they start to so bing bang boom, overnight you get these colonies that form that are actually they're visible to the naked eye, and they're actually after that first incubation period they're about 3/4 of the size of the final bean that you would buy in a bag.
Wow.
Yeah.
I'm taking notes.
Good, I'm glad. This is important.
So I've crowned you basically coffee queen.
Well, thank you.
Because
that means a lot coming from you.
My knowledge pales in comparison to this. I was but a little baby. I had just scratched the surface of the bean.
Oh, you shouldn't do that without gloves.
Oh no. Oh no. Huh?
It's probably fine.
You'll be okay.
It's probably fine.
Holy moly! Thank you for sharing your knowledge.
You're welcome.
Holy moly!
Deep eldritch knowledge now
Did
that
that
my
answer your
mind
question?
could not take.
Yes.
Okay. Okay, good. I'll go back to comms now.
Okay.